recording. This is your first time watching video. You're going to write down every time I say one of the attendance words throughout the lecture, or attendance phrases. And also you'll take a screenshot when I say one of those attendance words and attendance phrases. And at the very end, send in a list of those words with their accompanying screenshots, and also include one question about the material covered in the lecture. And the first word for attendance will be photosynthesis. So we should take a screenshot. So I should see the dark room with a lightly hallway lit up in the screenshot. But anyway, that's it for the first word. We've got about 30 minutes until lecture starts, so you can skip ahead in the video about 30 minutes. I like to get the recording started early. That way if something goes wrong, I can catch it because I'm paying attention to it. But once we get started with lecture, if it stops recording, just don't notice. Let's see. Excuse me. So are you normally an early riser, or is it just for this class, or for some other reason?
Usually am early riser, but fucking tired.
I get that. How old are you?
20 years old.
Yeah, I guess I was an early riser too. Only because I had to be to, the Marines. Or actually, that's not true. Depends. We had night crew and we had day crew. So obviously, if I was night crew, I was not an early riser.
Yeah, I love it.
But then once I got out of the Marines, man, I was night owl and an early or late riser for sure. Right? I used to bounce at different bars in Tallahassee, and we didn't get out there until like 3 or 4 in the morning. Sleep in until about noon. Grab something to eat. Go hang out in the pool until it's time to get ready and go back to the bar. It was a fun existence. Not a good one, but fun.
Sounds like a fun one.
It was a lot of fun, especially after 5 years in the Marines. And going from that life to just a life of nothing but fun.
What was your, what did you do in the Marines? What was your rank?
I fixed jets as an aviation electrician for F-18s.
Oh shit, that's cool.
Yeah, that was a good MOS because our MOS school was in Pensacola Beach. That's like where you learn your general aviation electronics, and then specifically to learn how to do aviation electronics on F-18s, and they sent me to school in Oceana, which is Virginia Beach. So I went from beach to beach, and then because I was working on jets, it gave us a lot of opportunities to visit places because one of our jets broke down, we'd have to go fix it. So that's how I got to see Iwo Jima. We were in Okinawa and one of our jets broke down, and they sent a few of us to go fix it. We were there less than a day, but it was still cool to see it. 8. So,
So exam 2 open right now, or?
yeah.
Okay.
Morning. Morning. Yes, I'll send you the email. I saw the link in the email. I saw it, but I haven't opened it yet. I'm still catching up from the weekend.
That's all.
Green, red, blue, white. Excuse me. All right, less than 2 minutes. Excuse me. Go ahead and start attendance. I'll take the sheet pulled up. Excuse me. Yes. All right. Let's see. Brumfeld, not here yet. Champion is not here yet. Gorby is not here yet. Gross is not here yet. Gunther is here. Randall Hill is here and was the first here, so we'll get a little extra credit for that. The other Hill is not here. Jackson, not here yet. Keys, not here yet. Perez is not here yet. Sigmund, I think, is here. Yep. Are you wearing WVSU stuff?
Yes.
All right. And WVSU here. Slaughter, not here yet. Smith was here. In WVSU here? No, just checking. Stover is here. Waiting for WVSU stuff. Tompkins is here. Is that WVSU? No, just checking. The Nest is here. WVSU here. Watson? No Watson yet. Young is here. Got WVSU stuff on? Or? Okay. Zwybeck is here. Doesn't look like WBSU stuff. Lawrence, not here yet. Carter is here. Did I miss anybody? It's 8 o'clock. We give people one more minute to show up before we get started. Does anybody have any questions in the meantime? One question you guys missed that was asked before you got here. Somebody asked, oops, guess it'll help if I pull it up. Somebody asked, based on the announcements, oops, when the exam is open, and the exam is currently open. Oh, where did it go? There you go. Yeah, so exam is open, ready to go. Anybody else? Well, it's 8:01. Guess we'll get started. Excuse me. So the usual announcements, reminders that attendance is mandatory. It's great. I'm not going to explain how it works. I've been doing it all semester. I'm sure you know how it works. October 12th is the last day of the half, so it's October 13th. I have to turn in midterm grades, including your announcement or your attendance. And remember, attendance is a full letter grade. So if you have any absences and you feel like making up for them, send me an email. I give you a list of which days you were absent, then you can watch the videos for the days you missed. Any questions about attendance? All right, first word for attendance would be pumpkin seed. Because for some reason, October 1st is National Pumpkin Seed Day. For extra credit, feel free to write any kind of biology papers about pumpkins. Where are they native to? What are their nutritional values? This room is haunted. It's, what in the world? That's weird. Weird timing too, okay. Anyway, yeah, so that's the first word for attendance. Lab today, that's as usual, right? We're gonna be here at time at 9. I'm gonna lock the doors. No one's coming in after that, except that, we have 2 students that come from another section that's a whole different story. But anyway, labs at 9, we got time. Make sure you wear pants or closed-toed shoes. I'm okay because I won't be doing any experiments. I'll just be walking around talking to you guys. Speaking of which, well, cross that bridge when we get there. As usual, I shouldn't say as usual, this is a more complicated lab in a sense, right? Because this is the lab you have to write a lab report for. So it's like a one-stop shop. Everything you're going to need for this lab, if you go to the course announcements, go under labs. So I've got the video of the pre-lab lecture that we did last week. I've got a link to the lab itself. I've got a link to the extra credit lab you can do, which is what I was getting at in this bullet point. If you missed points because you didn't do the pre-lab, or for whatever, or even if you just didn't even miss any points, just want extra credit, you can do that online virtual version of the lab. And those points will get applied to your missing points. So any questions about lab today? All right, pre-lab 7 is due on Friday. This time it's just 2 sets of video questions. And then the introduction questions, there are no hypotheses this time. This is more of a demonstration and more of an exercise than a true experiment. Because for the most part, you're not testing any hypotheses. So that does make it a little bit easier. It's also a virtual lab. So it's 100% online, meaning you need to bring your computers or whatever devices you're using to lab next week. Also meaning, if you happen to do it early, at least you'll get some extra credit probably. But anyway, you can possibly get some extra credit if you do it early, and also you just might be excused from coming to lab next week. If you turn it in this Friday, for example, let's say you turn in the pre-lab and the full lab on Friday, and then I grade it, and you got full credit, then I'll just say, all right, well, there's no need for you to come in next week. So, any questions about the pre-lab? All right, again, you can still retake exam 1, unlimited retakes, do it as many times as you want, open book, not timed. It's your highest grade is the one that counts. So just again, don't forget to let me know when you're done so I can go in and change your grade, because otherwise I have no way of knowing. Any questions about that? Okay, exam 2 is now open. It covers what we just finished, right? Yesterday was the last day of lecture for the stuff that's going to be in exam 2. It's due on Sunday, October 15th. You can do it anytime you want. If you want more than 50 minutes to take it, you need to contact me so I can proctor your exam so that we can extend your time limit. So also, again, that's the midterm, right? So the day after that is when I have to submit your midterm grades. So if you're concerned about that for any reason, now's the time to start working on it. But I would also say this, if you're concerned about it, don't be that concerned about it. It's just to let you know where you're standing. It's nothing permanent. You could have an F at midterm and somehow pull out an A at the end of the semester. And as far as your GPA and your records are concerned, the only thing that ever shows up is that A. It's not like anybody can go back and see what you had in midterms. Or they, I don't know, maybe they could if they really wanted to, but no one does. So that's it for announcements and reminders. Any questions about that? All right, so again, as a reminder, we are done with everything from exam 2. So everything we start talking about from today on is going to be on exam 3. Hopefully you remember exam 2 is basically, but not fully, but basically about one property of life, which was energy, right? This transformation of energy. What we're starting now, basically, but not exactly, but mostly, what we're going to do for exam 3 is reproduction. Remember, that's another one of those properties of life. You're going to learn how things reproduce at the cellular level. So I'm not talking, well, we'll talk about this a little bit, but obviously, when a lot of people think of reproduction, they think of a male and a female, a human, for example, sperm and egg get together, eventually you have a baby. That's not necessarily the kind of reproduction we're talking about. We will mention that a few times, but we're looking at reproduction at the cellular level, at least for this chapter. Really quickly, we'll do this little guessing game a little bit because we're a little bit ahead of schedule, but we need that because towards the end, those last chapters, they're not necessarily hard, but they're slow. So it's good for us to be a little bit ahead of the game. So here's a picture of a woman, and these two pictures go together. Does anybody know what this is a picture of? Hopefully you recognize that. Shouldn't be hard, right? You've seen these things before or seen a picture of that before. What is that?
Is it DNA?
DNA, yeah, it's a molecule of DNA. Thank you. So if you happen to send in the extra credit words, remind me that you got that, get a little bit of extra credit. Yeah, it's DNA. And they put these two pictures together. Obviously, a DNA molecule is not that thick. Like if this was to scale, it wouldn't be that thick. You wouldn't be able to see it. It would be so thin. But the point that your old textbook was getting at here was that one strand of DNA is about 6 feet tall. So, it would be about, well, I don't know how tall that person is, but it would probably be, a little bit taller, I would assume. But anyway, that's what it's getting at. How about this? Does anybody know what picture, what this is a picture of? Hopefully you've seen this before. I'll give you, can eliminate one thing. It's not boogers. It just kind of look like it, but does anybody know what those are? And I want to remind you too, I've been saying it all semester, but not saying it enough. I prefer to have a conversation instead of me just talking at you. And I love wrong answers because if you're guessing something, probably your classmates thought it, and if it's wrong, then I can work on that and say, oh, I see why you think that, then I can explain why it's wrong and give you the right answer. So please, when I ask you questions, don't be shy to answer, especially if it's a wrong answer. That's actually more helpful for me than a right answer. So anybody know what those are? I love that. Bacteria, that does look like bacteria. And actually, it would be almost nearly impossible not to tell the difference between bacteria. And so that being said, I'll give you another hint. Well, no, that wouldn't be a good hint. Yeah, so these are things that are in your cells, but not in bacteria cells, at least not in that form. Any other guesses? And then also think about what we're talking about here, cellular reproduction. Yep. So we're talking about reproduction at the cellular level. That's okay. These are chromosomes. So almost the entirety of this chapter 3, exam 3, in one way or another, is going to have to do with chromosomes. Whether we're talking about how they reproduce or how they recombine, stuff like that. So chromosomes is a big thing for this exam. This one I'm not even going to ask because it's almost impossible to guess. Obviously, we're looking at an X-ray with some lungs, and that's a tumor. So by the end of this chapter, you're going to have a very basic understanding of how cancer works. And obviously, that's a very relevant topic because unfortunately, you or somebody you know either does have cancer or will have cancer, right? It's a very common disease. So it'd be good for you to have the basics. That way, if unfortunately you'll ever have to do any research on any particular type of cancer because it's affecting your life, you won't be starting from scratch. Just a reminder, a lot of my PowerPoints are based on the old textbooks, so I kept some of the stuff, including these pictures. Also, and for this chapter, I included this thing. At the beginning of every chapter, that old textbook started with something called Biology and Society, where it would give you like a relevant news story to that chapter. And this one includes something called parthenogenesis. I'm not going to talk about this, but if you download the PowerPoint, you can read into it. I'm obviously not going to test you on it. This is from the old textbook. But there is some ways. Has anybody ever seen the first Jurassic Park? And you remember how the dinosaurs were just all female, and yet they still reproduced? Well, that happens in real life. And it's not that uncommon. It's uncommon enough to where it makes the news, but a lot of times females, depending on the species, will basically clone themselves and have babies or offspring that are a clone of themselves. So again, for extra credit, you can write papers about that. Like obviously, here's one example: a female zebra shark. But there's other examples. You can look up and see what kind of species are known to do this, or which type of species are known to do this more often than others. And if you were to look it up, that's the word you would use. Parthenogenesis. So females basically clone themselves. Now, cloning themselves is actually not that uncommon outside of the animal kingdom, right? So for plants, for example, and we'll talk about that in this chapter. So anyway, yeah, there you go. This chapter, it's going to be a little bit longer and more complicated than what this shows, but it's broken down into two main parts, which is the first one is what cell reproduction accomplishes, and the cell cycle and mitosis. I want to make very clear here, because some people like to really follow along with a lecture and the textbook together. I'll let you know that this is really based on the old textbook. So the order that I'm presenting things is going to be a little bit different than the order that it's presented in the textbook. I don't want you to be confused about that. So here we go. Reproduction. You need to know what reproduction is. When it is, or it may result in the birth of new organisms. Like I said earlier, I think when people think of reproduction, that's usually what they think of. You know, having babies, for example. But more often than that, it involves the production of new cells. And that's what we're going to be talking about mostly, is that second bullet. And that is obviously more often. Like if you have reproduced, any of you have reproduced in this room, if you raise your hand, like I've reproduced. Anybody else? That was a trick question, because the answer is yes, you've all reproduced. You're reproducing right now as you're sitting here. You're going through reproduction. So yes, again, the first bullet point, as much as we think of reproduction as that way, more often it's the second. You are literally reproducing. You're not creating a new organism necessarily right now. You might be, I don't know. But you're definitely creating new cells. So that's what reproduction is. You need to know that. I might ask about that on the exam. Cells undergo two types of reproduction. Either way, it's called cellular division, so that's another word, or cellular multiplication too, which is a funny thing because in math, division and multiplication are kind of like opposites. In biology, in this case anyway, we're using it interchangeably. But either way, what you're going to end up is with two daughter cells. Right? You're going to start with one original cell. You'll end up with two daughter cells. And under one type of cellular division, we'll talk about the other type later, and I'm going to get some names to these later. Right now, this is an introduction. In one type of cellular division, again, you start with one cell, you end up with two, and those two new cells are genetically identical to each other and to the original cell. And that will be important later in the semester, or later in the chapter. You don't necessarily need to write it down now if you're a note taker, because I'm going to bring it up again later. And I'm going to give you a word that you're going to need to associate with this, but for now, this is just the introduction. Any questions so far? Okay. So before a cell can reproduce, a lot of stuff has to happen. But what we're focused on in this chapter, mostly, we'll talk about some other stuff a little bit, but mostly this is the story of the chromosomes, which is why we have that picture of the booger/bacteria looking chromosomes that I showed you earlier, right? It's all about the chromosomes, at least as far as we're concerned. Obviously, when you think about a cell dividing and reproducing, there's a lot of stuff going on. But again, for us in this 100 level class, almost entirely we're going to be focused on the chromosomes, which should tell you for the exam, most of the test questions are going to be about that. What are the chromosomes doing? Right? There will be some exceptions. We'll talk about it when we get there, but that's mostly what we're talking about. This might be a test question too. Like, what does reproduction matter for any organism? Well, for a multi-celled organism, we use it to replace damaged or lost cells. So that's what I was saying right now, as you're sitting here, you are reproducing. You are replacing damaged or lost cells. And you might think to yourself, well, I haven't been injured recently, so something's damaged. But it doesn't necessarily need to be that kind of damage. Sometimes your cells just get old and die. Therefore, they're damaged. Therefore, they need to be replaced. Growth, obviously. Now, a lot of us are done growing in this room. But, well, that's not true either. Even now, right, you still have fingernails growing, hairs growing, things like that. But yeah, growth is another big important role for reproduction. And of course, actual reproduction is what we use cellular reproduction for, right? So if we need to create a new organism, we're going to use cellular reproduction for that. In different ways, obviously. As you can imagine, a human is going to use cellular reproduction to produce a human. Different than a bacteria is going to use cellular reproduction to create another bacteria. So any questions about that? All right. Here's some pictures from the old textbook, some examples. Here's cell replacement. So one of these kidney cells this is a kidney cell that is in the middle of splitting, right? So it is in the middle of cellular reproduction. We're going from one cell to two cells. You just need more kidney cells in this example, so we're having more of them. This is a great example of growth, right? So at one point, everybody in this room started off as one cell. And then we split into two cells, and then 4 cells, and then 8 cells, and 16, 32, 64, so on and so forth. And that's just what this is a picture of. Right? These cells are still splitting. Here's an example of asexual reproduction, which I haven't really gotten into yet, but we will on the next slide. But this is basically the basic, like the really most basic sense of cellular reproduction. But yeah, so some organisms can reproduce that way, like this amoeba. If you're a single-celled organism, then cellular reproduction is obviously how you're going to reproduce, right? You're going to basically split yourself in half. There are some exceptions to that, and we're not even going to talk about those exceptions, but generally speaking, that's the case. Here's another famous example, I think. I don't know if you guys know this, but if you cut the arm off of a starfish, for example, the arm will grow a new starfish, and then the starfish that got its arm cut off will grow a new arm. Another thing, if you were to get a sponge, like not a cleaning sponge, but a real live sponge, you squish it through like a sieve to where you just broke it up into thousands of pieces. Each one of those thousands of pieces would grow into a new sponge, well, the ones that survived. And then, of course, some people are very familiar with this. You can grow a clipping. So if you get a clip of a plant, some plants are more acceptable to this than others. But anyway, if you get a clip, put it in some water, sometimes some extra hormones, it'll start growing roots, and now you have a new plant. As a matter of fact, I read recently an article about people stealing like this. So people are going to like Lowe's and Home Depot, and they're not stealing the whole plant, but they'll steal a clip of it, take the clip home, propagate it, and now they've got a whole plant. I forgot the word for it. It's not a biological term, it's more of a society term. But anyway, those are just some examples. Any questions about that? All right, so looks like my slides were out of order. I should have talked about this first, but here we go. Asexual reproduction. Again, there's 2 types of reproduction. This is one of them. So you need to know the differences between this one and the one I'm going to teach you later. Asexual reproduction. So this is non-sexual reproduction in a way, right? That's basically the same thing. Obviously, if it's asexual, if it's non-sexual, there are no egg and sperm required. Again, the parents and the offspring, like I mentioned in the previous slide, have identical genes, and that'll definitely be a test question, right? So in which type of reproduction are the parents and the offspring genetically identical? Asexual. Again, like I mentioned earlier, asexual reproduction, even though we think of it very often as how single-celled organisms create new organisms, which is true, but it's also how multicellular organisms, such as ourselves, grow, repair, and in some cases, like I showed you in the slide earlier, which should have been after this, this is also how some multicellular organisms can create new organisms, right? So again, cutting the arm off of the sea star, or getting a part of a plant and regrowing it. I put others. I doubt anybody's gonna say anything because I know you guys are really quiet, but do you guys right now off the top of your head know any other examples other than the sponge one that I talked about in the previous slide? Okay, I didn't expect you to, but that would be a good extra credit topic if you want to write a paper about that. What are some other examples of how multicellular organisms can use asexual reproduction to reproduce? And of course, other than parthenogenesis that we talked about earlier, which is also slightly different than what we're talking about here. So any questions so far? All right, there's a particular type of cellular division that happens for asexual reproduction, and you're going to need to know this too. So we get another test question. Which of the 2 types of cellular reproduction uses mitosis? And the answer is asexual reproduction. So mitosis is used for asexual reproduction. That will be a test question. Now, granted, most of the test questions are going to be more about mitosis, like in what stage, just to skip ahead a little bit. It has 4 stages, at least that's how we teach it in this class. So there'll be a lot of questions like, in which stage are the chromosomes doing this? And which stage are the chromosomes doing that? But yes, there will be at least one test question where you have to link mitosis with asexual reproduction. And if you've already done the pre-lab, then you already know that, right? Because you've already answered those questions. So again, Mitosis is how single-celled organisms reproduce because they're only one cell, right? This is part of asexual reproduction. And it's also how we, multicellular organisms, grow and maintain ourselves. So again, I know I'm saying this a lot, I'm just beating a dead horse here, but as you're sitting here, you are going through asexual reproduction. You are going through reproduction. You are doing mitosis as you're sitting here. Which is another good test, another good question for extra credit if you want to look into it. Like how much, like what percent of your cells right now are going through mitosis? When you do next week's lab, that answer will be, but that question will be answered sort of, but it's all virtual. So, don't take my word for it or the lab's word for it. You can look it up and find other sources. So any questions about mitosis or asexual reproduction? Because for now, that's the end of the asexual reproduction discussion. All right, the next word for attendance is going to be coffee. Because today is also, I think, National Have Coffee with a Cop Day. Yep, I don't make these up, I just report them. Have Coffee with a Cop Day. I'm hoping that doesn't happen to me, nothing against cops, but I'm a little bit busy today and I'm sitting with a cop, that means I'm either in trouble or getting investigated or interviewed to get someone else in trouble. So let's hope that doesn't happen. Anyway, so now we're moving on to sexual reproduction. Again, this is what most people think of when they think of reproduction. Like outside of biology class, when someone says reproduction, this is usually what they're thinking of. Your textbook, the current one that we're using, breaks these down into two different chapters. So there's asexual reproduction and then there's sexual reproduction. So again, what we're doing, what I'm doing, is kind of combining the two in a different order because I'm basing this off the old textbook. Now the facts are the same, the stuff you need to know is the same, with some exceptions, and I'll bring those up when we get to them. But again, I just want to remind you that. So if you're following along in the textbook, I don't want you to get confused because we're going in a different order. It is on purpose. Anyway, if asexual reproduction, if you remember, that is without sperm and egg, well, the opposite is true of sexual reproduction. You do require sperm and egg. You require them to come together in a process called fertilization, which we're going to talk about later. Another term you need to know is gamete. So when I say the sperm and egg need to come together, well, the sperm is a gamete, the egg is a gamete. It's a different type of cell. So sperm and egg are called gametes. The production of these gametes is done in something called meiosis. So there's mitosis, which we talked about earlier, which is for asexual reproduction, and there's meiosis. Meiosis is how we produce those sperm and egg. So to be clear, not that this is important for the exam, but just so you understand completely, if we're talking about creating a new multicellular organism, like a human, it involves both meiosis and mitosis, right? But we're specifically talking about the sperm and the egg, that is only meiosis. So again, like right now, probably you are going through meiosis, especially the men. So you're producing sperm and you're producing eggs. Those will eventually, maybe, depending on how your life goes, those will come together with the other gamete, right? And at that point, again, like I said earlier, then you'll have a single cell, and then that thing will go through mitosis, right? But the process starts with meiosis. So you should associate sexual reproduction with meiosis. Any questions? Excuse me. So again, in summary, even though we're going to talk about it more in detail, this is kind of an overview. There are two kinds of cellular division in the lives of sexually reproducing organisms like us. Like I just said, there's mitosis for growth and maintenance, and then meiosis. So it starts with meiosis, because that's how we produce the sperm and egg. Then those two things come together, and then everything after that is going to be mitosis. Right? So again, we start as one egg or one cell, then just two, or go to 2, 4, 8, 16, so on and so forth. That's mitosis that's making that happen. Any questions? All right, so now we get into some different information before we go back and talk about those things. So again, eventually we're going to talk a lot about how mitosis works and how meiosis works. Before you can understand that, you need some background information. Sort of like when we did photosynthesis and respiration, right? When we talked about photosynthesis in the last chapter, before we could get into those two stages of photosynthesis, you needed to know how sunlight energy worked and how photons worked and how pigments worked, all background information. The same is happening here. So let's learn about some chromosomes, because remember, like I said, when we're talking about this process, what we're focused on in this 100 level course is how basically what's happening with the chromosomes. So before we can talk about what they're doing, let's talk about them in general. So eukaryotic chromosomes. So to remind you, eukaryotic, that means us, right? So these are more complicated organisms, plants, animals, fungi, things like that. Prokaryotic, to remind you, would be things like bacteria, simple single-celled organisms. Which reminds me, for the most part, I'm not even going to talk about prokaryotic cells. For the exam, we're not going to talk about prokaryotic cells. So, excuse me. Anything I'm teaching you in this chapter, just remember this is only about eukaryotic cells. So if you want to, for extra credit, look up, right, well, what about prokaryotic cells? How is this different than how prokaryotic cells are? Anyway, let's talk about these eukaryotic cells. Most genes are located on chromosomes in the nucleus. You need to know that for the exam. Now, I put a little star there to remind myself that your textbook does talk a little bit about prokaryotes for this slide, but you don't need to know what your textbook says. You can read it, that would be great. I might have some extra credit questions on it. But for the exam, for this 100 level course, I want to keep it focused on eukaryotes. But yeah, most genes are located on the chromosomes that we find in the nucleus. I mentioned this in chapter 2 when I first introduced you to nucleic acids, right? When we talked about the biological molecules, you knew that then, and now I'm reminding you again. Actually, same thing with chapter 3, when you learned about the different parts of the cells, and you did learn about the nucleus. So yes, most genes are located in the chromosomes in the nucleus. There are some other genes located in your mitochondria, which is not important for this chapter or this exam. I just want to point that out so you don't forget that. So your mitochondria has its own genome. So all this reproduction we're talking about, this meiosis, has nothing to do, or mitosis, has nothing to do with those genomes. Same with chloroplasts, right? So plants have mitochondria and chloroplasts. So those chloroplasts have their own genomes. That's only just to remind you of that. So to keep things simple, we are only focused on what's happening in the nucleus. That is also a good extra credit topic. I don't even think your book goes into it, but you could look into like, all right, how do mitochondria reproduce then? Because you're going to learn how cells reproduce. Well, how does the mitochondria reproduce with their genes, their chromosomes? How do the chloroplasts do it? All right, so going back to these genes, these chromosomes that we find in our nucleus, each chromosome contains one long DNA molecule. So like one chromosome is just a long molecule. Of course, it's usually twisted up and have different shapes, but generally it's just one long molecule that has thousands of genes, which is another good extra credit topic if you want. As you're going to learn later, we have 23 pairs of chromosomes, humans do. Every species is different. And each one of those chromosomes is a different size. And as you might imagine, generally speaking, the larger the molecule, the more genes it has. So you can look that up. How many genes do we have in total? How many genes are on the average chromosome? How many genes are on each chromosome? You can look all that up for extra credit if you want. So like I said, we happen to have 23 pairs of chromosomes. Each species has its own number of chromosomes. The next slide I'm going to show you is an example of some species and their different numbers of chromosomes. You don't need to know it. But then before we do that, let's give you one word that you do need to know. One, because I'm going to use it a lot. And two, because I might ask you what it means on the exam. Genome. A genome is a cell's complete complement of DNA, at least in this context. Now, if you were talking about you sending off your spit sample to 23andMe, in that context, the genome is all of your, right, the complete set of your DNA, right? That's what we're talking about if you were looking at it that way. Or if you were doing a study to determine which genes are responsible for the heat in a hot pepper, right? Then you might take thousands of peppers and look at all their genomes to see which genes or which genes are expressed more in the hotter peppers and which genes are expressed less in the cooler peppers. And then from there you can determine which genes. But in that context, when we're talking the genome, then we're talking about the whole species. So just as a reminder, There are different ways of using the term genome, but in this chapter, since we're talking about cellular reproduction, we're talking about a cell's complete complement of DNA. Later in this chapter, and not this chapter, but this section, we're going to talk about your entire genome. In that case, we won't be looking at one cell, but we'll be looking at you as a person, as an entirety. Any questions about this slide before I show you some pictures or a graph of all these different species and how many chromosomes they have? All right, there you go. Just an example. You don't need to know any of this. I'm not going to ask you any of this on the exam. If anything, I would give you this graph and then ask you a question about it. Like, oh, how many chromosomes does a duck-billed platypus have? And you look and say, oh, it's got 54. Now, you might also remember that I said humans have 23 pairs of chromosomes. And that's still correct. But notice I said 23 pairs of chromosomes, which means double that, we have 46 chromosomes. Which I find interesting about this is that, and there's no pictures of it, but here we have, at least on here, the animal with the smallest amount of chromosomes is this deer. And I usually think of a deer as a larger animal. Meanwhile, the one with the largest amount of chromosomes is this rat with 102. But again, you don't need to know any of that. This is another great extra credit paper. Pick any organism you can think of and any eukaryotic organism you can think of that you're curious about and find out how many chromosomes does it have. How many chromosomes does a pumpkin plant have? How many chromosomes, we're looking at a koala, how many chromosomes does a wallaby have, or a kangaroo? You can look into any of that if you want. Plants are interesting, and I might talk about it briefly later. I'm going to ask you any questions about it. But just so you know, like I said, humans have 23 pairs of chromosomes. A lot of these things probably have pairs of chromosomes, but plants, they get a little bit tricky. They can have sometimes 3 sets of each chromosome, or sometimes 4. But again, that's beyond the level of this course, and you can look into it for extra credit if you want. Any questions about this slide? All right, let's talk about what chromosomes are made of. This will be important later in the semester. Because to understand how they work, you really need to understand how they're put together. That sounds familiar too, right? Structure and function. It's the same thing. Chromosomes are made up of something called chromatin. Obviously, if these things are located inside the nucleus, they must be smaller, thinner, so that they can fit. But what's a little bit less obvious is that they're actually a lot longer than the nucleus. Like I said, if you were to have one strand of DNA, it would be about 6 feet long. So imagine that. Imagine having 46 of those things all squished into a tiny nucleus in just one cell, and then all your cells have those. So you can imagine it must be very thin, and it must be very squished to get it in there. And again, structure and function are very important here. To learn how they work later, you need to know how they're put together right now. They're made up of, chromatin is made up of DNA and protein. So even though we're focused on the DNA, like for this whole exam 3, when we're talking about chromosomes, we're almost always going to be talking about the DNA because that's what we're focused on for this 100 level class. But it also has proteins incorporated into it. And those proteins are important and will be important later in this exam 3 section. Well, this is module 2, module 3? Yeah. Anyway, it will be important. Again, when you learn how these things work, it'll be important to understand and remember the fact that they also have proteins. These proteins organize the chromatin, so they're not just hanging around, they're there to help organize it. That'll make a lot more sense when I show you a picture of it. Later, you'll see how the proteins organize the chromatin. And this will be important later too, they help control the activity of the genes. So yeah, you've got all these genes, but think about it. Like we talked about earlier, much earlier, genes don't need to be constantly working and making stuff, right? Sometimes you need what they produce, sometimes you don't. So we have to have ways of turning them on and turning them off. That's what the proteins help do. Like I mentioned earlier, from that bullet point, if you were to fully extend one DNA molecule, the average DNA molecule, it would be about 6 feet long. Which I don't think will be a test question, but it is a question on one of the labs that you're going to have to do. Not this week's lab, I don't think, but maybe the week after that. As your cells prepare to divide, whether it's in mitosis or meiosis, the chromosomes, what does it say that? I could probably get rid of that. The chromosomes are coiled up. So I need to fix that. It's not that like, as they're preparing to divide, that the chromosomes are like all uncoiled and like, oh, we're preparing to divide. Well, no, that is true. I'm trying to think about how I want to put this. They do coil up, but yeah. Later in this chapter, I'm going to get very specific and tell you when and how it happens. So it's all a matter of context. I hope I didn't confuse you. Because in my brain, I'm thinking of, all right, are we talking about the beginning of mitosis or some other time? So don't worry about that for now. But yes, they do have to coil up before they can divide. And I'll talk about why later. And when I talk about why, hopefully that'll be easy to remember the fact that they coil up. When they do coil up, they form compact structures that you can see with a microscope. So before that, you cannot look at a microscope picture of a cell and see a chromosome. You can see a bunch of chromosomes all squigglied up together, but you can't pick out one chromosome because they haven't coiled up yet. I'll show you a picture of that on the next slide. So any questions about this slide before I show you the next slide? I'm going to slow down because I see people taking notes. All right, so here's a picture of probably an onion cell. I'm not sure. The old textbook, this is from the old textbook, not the new textbook. The old textbook just says it's a plant cell, and I already know that because it has this square shape because it has cell walls. But you see here, this is the nucleus of a cell that's not about to divide. So you can't see and be like, oh yeah, there's the chromosome, right? There's just a bunch of squiggly mess. All that DNA is uncoiled, and I'll talk about why that's important later. Here, they have coiled up, so you can see. Now, granted, I can't look at that and say, okay, there's chromosome number 1, and there's chromosome number 2, there's chromosome number 3. Right? I can't do it that way, but I can look at that and see, I can see the chromosome structure as opposed to a big squiggly mess. To me, these pictures look like a bunch of purple sausages, I think. Of course, they don't naturally have that color. To look at them in a microscope, you have to dye them first, but you don't need to know that for the exam either. So the next word for attendance will be sausage, since that to me looks a little bit like sausage. So any questions? All right, let's go back and talk about those proteins a little bit. So remember, chromosomes are made up of stuff called chromatin. Chromatin is about half DNA and half proteins. So again, like we said, those proteins serve two functions. One of the functions is to control the gene activity. The other function is to help organize the chromosomes themselves, and that's what we're talking about here. The DNA is packed into an elaborate multi-level system of coiling and folding. So it's not just a big squiggly mess like you see, well, even then. It's not just a big squiggly mess, it's organized. They coil up, they wrap around. And these proteins help them do it. These proteins are called histones. I might ask you that on the exam, like the definition, like I might ask you what a histone is. More than likely, though, I'm just going to use that word in a question. So if you don't know what a histone is, the question won't make sense to you. So what is a histone? Histones are the proteins that are used to package the DNA. So again, talk about what we talked about on the previous slide. When we're talking about chromatin, proteins do two things: control the activities of genes and organize the genes. Well, these are the ones that do the organizing, right? These are not the ones that necessarily control the activities of genes, but we'll talk about that later. Just for the sake of the exam, you should associate histones as the proteins that are involved in this organization, this packing and folding. This will make a lot more sense when I show you on the next slide. So these histones come together, the DNA wraps around them, like I'll show you on the next slide. And each time that happens, that's called a nucleosome. So I'm not going to talk about that right now. I'll just show you the picture to make a lot more sense. So here's a drawing of a DNA molecule, that double helix wound around itself. And then you can see here, those are wrapped around these histones. It's technically 8 of them. I'm not going to get into that, but it's basically forms like a little cube almost, a cube of round proteins, and it just wraps around and wraps around and wraps around. So each one of those little things where it's wrapped around 8 histones, that's called a nucleosome. And again, it isn't that I'm probably going to ask you questions about that. I might, but probably not. The idea here is that I'm going to use these terms in the exam and when I'm teaching you this chapter and later chapters So when you need to know what a histone is and what a nucleosome is, that way when I use the term, you know what I'm talking about. So any questions about this slide? All right, so another question you could ask yourself for extra credit if you want is here is this picture. I'm showing you each nucleosome has 8 histones. So you can look into it and say, are all nucleosomes like that? Is that just for humans or is that just for particular cell types? So you can look that up if you want. Also, again, we're talking about eukaryotic chromosomes. So then the other question you could ask yourself for prokaryotes, like do bacteria even have histones and nucleosomes? Or is this strictly a eukaryotic thing? Here's another picture. We're kind of zoom out a little bit more. So even though they've wrapped up, you know, the DNA molecules have wrapped up around those histones. In this case, they even got, since they zoomed out, they made it a little bit simpler. Here they show all 8 of the proteins. Here they're just showing it as one big blob. But anyway, that even coils up on itself and in that shape. And then that even loops up on itself in that shape. Right? So that when you're looking at a chromosome, this is what you're looking at. It's highly coiled up. Because again, you have that one DNA molecule out of 46, and it itself is 6 feet long. So imagine packing all that DNA into one nucleus, right? So you're going to need to coil it up. And again, later in the semester, or maybe this chapter, that'll be important. So zoom out one more time. There's both of those pictures combined into one. Again, these pictures are not from your textbook. So if you find that these are easier to help you comprehend what's happening than what you have in the textbook, you can get these PowerPoints on the WVSU online. So any questions about how chromosomes are put together before we move forward? Okay, let's talk about duplicating chromosomes. Because again, like I said previously, what we're focused on, even though a lot of stuff is happening, is we're focused on mostly, almost entirely, is what's happening with the chromosomes. So there's a lot of stuff going on in the cell, but what we're focused on is what's happening with the chromosomes. So obviously these chromosomes are going to have to duplicate themselves before you have 2 new cells, right? So that's what we're going to talk about first, is how they duplicate. So here we go. This is basically what I just said here. Before a cell can divide, all that DNA has to be copied. Because remember, especially if we're talking about mitosis, right? I said the parent cell and the 2 daughter offspring cells, they are genetically identical. So that tells you, obviously, we have to copy the DNA, and we have to copy it without any errors, speaking of which. And the DNA has to be copied. Even in meiosis, even though they're genetically unique, we still need to copy the DNA, and we'll talk about that in more detail when we get there. But yeah, so whether we're talking about mitosis or meiosis, the DNA has to be copied. And what happens when we do this is we then end up with something called sister chromatids. This will make a lot more sense on the next slide when I show you a picture of it. This is another one of those terms where it's not that I'm probably going to ask you, what is a sister chromatid? But I'm going to use this word a lot. I'm going to use it a lot to describe things when I'm giving you a lecture. And then I'm going to use it a lot in the exam when I'm asking you something. So it's not, again, not that I'm going to say, what is a sister chromatid? But when I ask you other questions, the term sister chromatid will be used, so you need to know what it is. Sister chromatids contain identical genes, and they're joined together at a waist called the centromere. That's another word where I'm probably not going to ask you what it is on the exam, but I will be using the word centromere a lot in the lecture to help describe things, and I will be using the word a lot in the exam to help ask things. So again, this is going to make a lot more sense when I show you the next slide, and then I'll ask you if you have questions. So you start with this single chromosome. It copies itself, right? A perfect duplicate. And what happens is it's still attached, right? They're still attached to each other. So technically, instead of having 1 chromosome, you now have 2 sister chromatids. And they are exactly the same. And what they're not shown on this picture is they're joined together right there at the centromere. So again, even though it's not shown, and the centromere is always in a different place. We'll talk about that later too. But here's where the centromere would be. They're attached together. So again, what we're looking at is 2 different chromosomes, but technically Since they're still together, we call them sister chromatids. They have perfect copies of each other. There you go. So any questions so far? Pretty simple stuff so far, right? So if we're talking about cellular division, obviously those things are going to have to be separated, right? You can't have them together. That would be useless. We're trying to separate them because we're trying to turn one cell into two new cells. So when a cell divides, the sister chromatids have to get separated. At that point, we just call them chromosomes again. So even though they're still identical to each other, since they're not together anymore, since they've separated, we just call them chromosomes. And again, it's not that I'm going to test you on the nomenclature. Just when I describe, like, all right, we have this chromosome, and it duplicates, and it becomes chromatids, sister chromatids, and then those things separate to two separate chromosomes, right? You just need to understand the terminology when I use it. And genetically, nothing has changed between the sister chromatids and the chromosomes. All we did was separate them, right? Those sister chromatids, again, are perfect copies of each other. But then once they're separated, we just happen to change the name. Right? They're no longer called sister chromatids, they're just called chromosomes, but they are still identical copies of each other. So there you go, it's pretty simple. Start with one chromosome, duplicate it so you have two exact copies, separate it, you still have two exact copies, but now they're full-blown independent chromosomes. Any questions about that? All right, that's where the easy stuff stops, sort of. The next stuff we're going to get to is slightly more complicated and definitely, in my opinion, more important. We're going to talk about the cell cycle. Now, in the context of this chapter, and even for the exam, the cell cycle is a little bit less important. But in the context of your everyday life and to truly understand how cancer, for example, how cancer works, you have to understand the cell cycle. Because as you're going to learn when we talk about cancer, cancer is a disease of the cell cycle. So to understand cancer, you need to understand the cell cycle. Let's get the introduction to this before the end of the lecture. So the cell cycle is an ordered sequence of events. Obviously, I'm going to be teaching you about each one of those events. It starts when the first cell is formed until that one cell finally gets old enough and big enough and splits into two cells. So it's almost like the life cycle it is. It's the life cycle of a cell. But instead of like for humans, you might think of the life cycle as when you're born to when you die. It's different for one cell. Here we're just talking about once you're first formed until once you yourself split up into two new cells. Of course, some cells die in the middle of the cell cycle. They never get through it. But yeah, basically the cell cycle is the life cycle of a cell. And in a sense, there is almost, unless it does die somewhere in the cell cycle, in a sense, they're almost like, you could almost argue that they're immortal, right? Because they don't necessarily die. They're created, then they go, and then finally they create two new ones. So it's not like they died or disappeared, they just became two. Then those two will become two. And then those two will become two, so on and so forth. There's two basic parts of the cell cycle, and you definitely need to know this for the exam. It's the interphase and the mitotic phase. So sort of like I suggested on the last chapter, and you can study however you want, but my suggestion is to learn the big picture and then start adding the details. So the biggest picture here when it comes to the cell cycle, the whole thing is interphase and mitotic phase. You can almost think of it like a clock or like a day, right? If you're talking about the day cycle, the cycle of the day, 24-hour period is broken down into two phases, right? You got nighttime and daytime. And of course, you can subdivide nighttime and daytime, but those are the basics, right? You got nighttime, daytime, dark and light. Same thing here. The cell cycle is interphase and mitotic phase. And we can and will break those down into different things. The interphase, there's different parts of that. The mitotic phase, there's different parts of that. You're going to need to know all that. But for now, the most important thing you know is there's interphase and mitotic phase. Learn and memorize that. Then learn about the different parts of interphase. Then learn about the different parts of the mitotic phase. Here's a picture of the cell cycle. I don't remember if this is the old textbook or your textbook, but again, if it happens to be different than what's in your textbook and you like this picture more, you know where to find the PowerPoint. But yeah, there it is. Here's interphase and all the different parts of it, right? Interphase and mitotic phase. So again, study and memorize that. Then once you know that we have interphase and mitotic phase, then start learning the different parts of interphase like G1 and S and G2. Then start learning about the different parts of the mitotic phase, which are not even shown here. But yeah, there we go. That's a great place to stop. So the last word for attendance would be blue, since I just had a lot of blue on this slide. And of course, the true last word for attendance is you asking me one question about the material. And that's it. We'll be back here at 9 a.m. to jump into the lab. We've already done the pre-lab lecture, so we're going to go pretty quickly, or start pretty quickly. So do whatever you need to do between now and then. If you need to have a drink, make a phone call. Thanks. All right. Yeah, let's go ahead and set things up. What's that? Yeah. Actually, you know what? I don't even know if we're gonna need that or not yet. We'll see. I wanted it just in case, but we'll see if it works or not. So we could do like group 1 here, group 2, group 3, group 4, group 5. Group 5, if we can, if we have room, if we could set them up over there. Yeah, because they have 5 different lights or 4 different lights. And if we put them up like this, then the one closest to the window is going to have more photosynthesis than the one farthest from the window. So that'll be messing up instead of color, which is where they're doing the experiment on. It'll also be dealing with light intensity. So we'll eliminate that by doing that.
Sure.
I'll show you. Let me help. Actually, you know what? We might be able to keep these here. And what we can do is keep them separated, so they're yeah. So it won't be perfect, but this will help block the white light from whatever color is there, So even that's going to be helpful. Light pollution is the thing for this. Let's see. So after the pre-lab lecture, we will come around and help you guys set things up, but so you're not going to necessarily do it on your own. There we go, different colors, yeah. So that's, yeah, okay. That's light intensity, respiration, photosynthesis, yeah. Yeah. Separate that. That's perfect. Very good. Oh, can you get some ice while I get some? No, do you know where it's near the core lab? Do you know where the autoclave machines are?
No.
Yeah, same, there's an ice machine in there.
Yeah, check it.
Thank you. One for there. One for there. There. Yeah. Here. Alright. Ruler here. Temperature and photosynthesis.
Do we have to drink from here?
Yeah, please. Yeah, make sure if you have drinks or food, you put it outside, put it up. So, respiration, photosynthesis. That. Let's see. One in the dark. This is gonna have one in the dark. That.